Good afternoon, everyone, and welcome. I'm Jack Van Horn from the University of Virginia, and it's my distinct pleasure to welcome you to this installment of our 2025-2026 Foundations of Biomedical Data Science seminar series. Our unique lecture series is made possible through the generous support of the UVA College of Arts and Sciences, the UVA School of Data Science, and through a grant from the National Institutes of General Medical Sciences. Since its founding in 2016, the Foundations of Biomedical Data Science seminar series has been at the forefront of exploring the transformative impact of data science methodologies, artificial intelligence, deep learning, and statistical modeling across diverse themes in biomedical and health sciences. Our theme that was selected for this year is focused on data science approaches and the study of autism spectrum disorders, where every Friday we'll enjoy presentations this academic year from a wide range of leading thinkers about the issues, promise, opportunities, and hurdles associated with research in autism spectrum disorders. This involves deep phenotyping, clinical assessment, advanced genomic, transcriptomic, metabolomic, and other omic methods, along with a wide variety of data analytic opportunities that they present. We are particularly excited for this year's seminar series program where biomedical researchers and computational experts will shape for us the domain of autism and provide insights into its quantitative analysis. This topic is very newsworthy right now and subject to much debate and possible misunderstanding concerning its causes and its treatments. Clearly, bringing data and novel methods to bear to understand the role of genetics, the developing brain, and the environment will be very important to explore. So we're very excited about covering this particular novel and topical area of research and investigation. And for today's seminar series lecture, I'm delighted to welcome our speaker, Caitlin Hudak, from the Department of Psychology at the University of South Carolina. Dr. Hudak received her BA in Human Development from the University of Chicago before launching her research career in cognitive neuroscience at labs at Carnegie Mellon University and at the Yale Child Study Center. She did her PhD in developmental psychology from the University of Nebraska-Lincoln and completed postdoctoral training at the University of Washington in Seattle, and now she is an assistant professor. And then was assistant professor at the University of Alabama before coming to South Carolina in 2022. She directs the brain research across development, or the BRAD laboratory, and is the steering director for the Carolina Autism and Neurodevelopment Research Center. She's an associate professor of psychology at University of Southern California, and she studies the brain mechanisms of autism and rare genetic conditions linked to neurodevelopmental disorders. She is an expert that connects biological markers, including EEG, ECG, eye tracking, and fMRI, to individual differences to understand basic science and extend them towards a precision medicine. In the spirit of our 2025-2026 Biomedical Data Science Seminar Series theme, her lecture today is entitled Rare Genes, Shared Pathways, Biomarkers of Brain Development in Autism, where she will explain for us that brain-based biological indicators, or biomarkers, could accelerate and leverage efforts towards clinical interventions for neurodevelopmental disorders such as autism and other intellectual disabilities. Yet there's a tremendous genetic and phenotypic heterogeneity to consider across and within these different neurodevelopmental disorder groups, even with rare genetic subgroups. And so we're really excited to hear from her today. As we always do, we are streaming this lecture live and for recording via YouTube. So if you're watching us on YouTube, thank you so much for joining us. Also, our specially selected 2025-2026 Biomedical Data Science Innovation Lab participants, as well as alumni, are encouraged to submit any questions they have via the chat feature in their YouTube sessions. I will synthesize those and ask them on your behalf during the last 10 minutes or so of Caitlin's lecture today. And with that, Caitlin, thank you so much. We are really eager to learn about your work and hear about your perspectives.
Thanks so much. What a great introduction. I am excited to share some of the work that we've done and are continuing to do, and hopefully entice folks who are new to the world of EEG to understand its utilities, and also share some of the links that we've made into a really truly translational nature of translational science and translational neuroscience specifically. I have a lot to cover, but I thought it might be important to start by just making sure that we are all on the same page about how we understand and diagnose autism spectrum disorder. And this is important to us because when we're thinking through using things like biomarkers, it's helpful to know how we're getting to the behaviors that we're thinking that the brain is going to capture for us. So with autism spectrum disorder, we we see these two sets of differences where our individuals are noted for having these differences in socio-emotional reciprocity, as well as having some challenges related to communication. And that can include nonverbal communication, as well as maintaining these relationships. Whereas they don't have to have the exact same criteria as you see on this right side for criteria B, but we see that our autistic individuals are going to have stereopities or kind of be stuck on repetitive motor movements or using repeated kinds of speech or using objects in one particular way or having other kinds of inflexibilities related to sameness or behaviors that you might see. So this might seem straightforward. We've written a very long and referred to a very long book called the Diagnostic Statistical Manual to understand, is this autism or not? But I want to really introduce you to a couple of different kiddos. And you can see this kiddo here we've seen multiple times. You can see why we are truly a developmental brain lab. And what's really interesting about each of these three kiddos is that they all have autism or suspected autism symptoms, but they also have intellectual disabilities, and to varying degrees. And if we start to drill down on some of the pieces that the parents are telling us are maybe the most important thing to try to track and understand, and that might be feeding into these diagnoses that we see, you can see that there's a variety of differences. Sometimes it's attention, sometimes it's communication. And what's really kind of neat from our perspective is that each of these three individuals have a different rare genetic condition or variant that is linked to autism specifically. So what we try to do in my lab is try to understand how all of these pieces fit together. But what's really important is that if you drill down even further, we're talking about very different levels of a person, right? So diagnoses or behavior, and mapping that all the way down to the genotype. There's a lot of interesting pieces in here, and hopefully I'll convince you today that using neuroscience in particular is a way to understand and maybe link between these different pieces. So I thought I would go ahead and just show you a little bit of what I'm hoping to share with you today about how we're using biological mechanisms to really push what we can do with our science and use it in this translational way. But to do that, we need to develop biomarkers that are appropriate and potentially think through not just putting all of our eggs into one biomarker basket, but thinking about how we can use multiple biomarkers and different computational approaches to understand patterns, while also being aware of that these are not static in time, that we need to account for maturation and development. And then hopefully I'm going to save enough time to share a little bit about the work that we do to truly improve inclusion in neuroscience research to make sure that we're seeing this progress for all autistic people, including individuals that have co-occurring intellectual disabilities. So it's important to think through how we're building the person and the brain. And when we think about these pieces, coming back to kind of our little rainbow model here, we're able to see things at this top level from a clinical diagnosis where you have a clinician or a provider who are providing this information. And this is not necessarily just neurodevelopmental disorders like autism and intellectual disability, but also thinking about co-occurring medical conditions that are very common, including gastrointestinal issues and seizures, which are things that we see in autism often. But then also thinking about how the things that we see, their observed behaviors and social behaviors in particular, are things that we can capture. Now, what's tricky is that a lot of these pieces at the top of this phenotype pyramid are less easy to address with medication alone. And often we're using more group therapies and other kinds of physical therapy, occupational therapy to really try to understand these behaviors. Whereas it's this lower biological level where we see a lot of pharmacological interventions as commonly being helpful. So when we think about translational science, we have this kind of framework in mind, where if we're trying to track and having early identification of autism or really any sort of neurodevelopmental disorder, then we need to be tracking as early as possible. And because having this early identification allows us to use different interventions, whether they're in person-based, in-person therapies or using appropriate pharmacological solutions. But what we're really trying to do is tease apart how all of these pieces then map back on to how our bodies are developing. And specifically in my lab, we focus on brain development. And using what we know about genes to understand, maybe that's even a way of understanding and early having this identification of potential elevated likelihood of autism. So from this translational perspective, we have used fMRI in the past. We currently are using a lot of EEG, as well as using translational models like our fruit fly to understand this basic science mechanism with this idea that by the end of my career, hopefully, we will have translated this into what we really see in our human participants and our clients and be able to develop those interventions, test those interventions, and implement them to then see how we're actually affecting this developmental change and working with a plastic brain to help improve the lives of our kiddos. So when we think about this, we have these, again, these complex kind of clinical diagnoses. They're often co-occurring with medical conditions. We have a slew of different behaviors that we can link back to autism specifically. And so we can use our neuroscience tools, including FNIRS, here to really start to tease apart part of this pyramid. But what's really exciting to me, even though I'm a cognitive neuroscientist that works with humans, is that there's all of these other pieces and that we really improved our scientific progress to use all of these tools, including induced pluripotent stem cells, really looking at the protein-protein interactions and understanding these at a cellular level with our partners in crime in more basic neuroscience labs as well. So if we take an example, one such rare genetic condition that's strongly linked to autism as well as hypotonia and macrocephaly is what's called CHD8. And this is a gene that helps control for and modify the way that our genes are able to kind of produce proteins. So this is a very strong and well-known link. And I was able to present with a group of folks a couple years ago at a conference, and we had Dr. Emily Newhouse, who spoke about what we see at the kind of diagnostic and behavioral level in individuals with CHD8. And Dr. Rebecca Moulay talked about what she's seeing in the mice and specifically seeing and showcasing this increased anxiety response, where I came in and spoke a little bit about the human and the fruit fly habituation, which actually mapped really well onto what Dr. Ellen Hoffman talked about from the zebrafish model, and seeing some kind of overlap there. And then Dr. Helen Wilsey spent some time talking specifically about how the proteins are localized within the nucleus, specifically in the brain. So what I'm trying to emphasize here is that we really need to be thinking through what kinds of systems we need to use to address different questions. So I think one of our kind of big tenets is that we don't necessarily need to rely on only the human work, but be interfacing as closely as possible with our folks in related neuroscientific fields. And there's certain models, which might be the human and might be other animal systems, that are going to address the different questions. And so making sure that we're using our specific tools to really understand and find where those links are across these other species and other domains as well. So we think that using kind of biomarkers in this way, where it's truly thinking lower into some of these cellular and protein levels and then upwards towards the actual diagnosis, is a way to understand why different autistic individuals might look different than others and where we might see some shared phenotypes. So the whole idea of this is if you have some heterogeneity, our goal is to get to this point where we have these distinct subgroups, which we can then use these biological indicators to help us understand how best to create these subgroups of autism. And we are not the first to do this. There's many amazing groups across the world and across the US that have been really working hard to create these biomarkers in a reliable way. And so I wanted to share just a little bit about what our kind of overall goal is. So we want to be creating these biomarkers with this notion of being ready for clinical trials, so that if you have a specific treatment that you'd like to test, and this might be something like a pharmacological intervention, using these biomarkers to kind of quantify how this clinical trial is going is one potential solution. And these are really meant to kind of showcase what is that clinical piece specifically. So what part of the biology maps strongest onto the clinical phenotype. And I'm not going to review all of these, but I think it's important to review the fact that being mindful about how you're planning to use the biomarkers is very specific. So for example, if you're trying to understand a predictive model, then you want to understand what biomarker is most likely to understand who will benefit from the treatment that you're planning to implement. So we have a lot of different uses for them, and being thoughtful and very explicit about how we're using them is important. But we're still very much in the kind of developing biomarker phase. So what does it mean to have a strong biomarker? So we're looking for something that's going to measure the subgroup of autism and specifically does not measure these other subgroups. And that might be neurotypical development. It might be autism without a rare genetic condition, for example. So how do we get to that is kind of the overarching question. So I'd like to give you a couple examples for how we've been doing this from what we call a genetics first approach. So this is where we start with individuals that have a known genetic condition. So for example, I'm going to share a little bit of our work with GRIN2B and SCN2A. And so we know that having a loss of this function, so having a variant that creates, interferes with both GRIN2B and SCN2A as their job as genes, has this cascade where we see specific mechanistic effects. And this is built on to then what we start to see at the cellular level, as well as how synapses are functioning specifically in the brain. And we know that these are also then linked to our kind of primary phenotypes like autism and intellectual disability, as well as actually diverging features of behavior. So in GRIN2B, we tend to see more spontaneous social communication, where we see less of that in SCN2A. And we've done some of this work to really try to understand, well, what are these different pieces and how is it and at what different level are we seeing those moments? But this is where we really see this opportunity for biomarkers to link across and to truly understand at a mechanistic level what is different in these two different groups. So we've decided and been using electroencephalography or EEG for many years now for a couple of key reasons. So most importantly, as I think this video kind of demonstrates, we can use these nets and the kind of getting it set and getting kids into this procedure is super simple. So this is work for my dissertation where I was working mostly with babies at the time. And aside from a couple of tears, usually the net was on and we're testing within seconds, which is fantastic. What's also really great is that we can really capture brain waves at all ages. So this is something that's very developmentally scalable. And you can see when we use these high density nets, we're recording from over 100 different channel locations every millisecond. So for those data scientists, we're talking about really, really time and space dense data. So this becomes a really interesting spatiotemporal problem or opportunity, let's say. So we've spent a lot of time focusing on attention as a specific feature because of how translatable it is. So attention is really meant to describe our capacity to allocate our attention and our resources to certain stimuli. And this is the example I like to share about novel stimuli. So if I give you a picture like this, you're probably scanning the image and it takes you a couple of seconds, but maybe at some point in time, you zero in on the thing that's not quite like the other, this pop-out effect. But what's really interesting about our attention system is that we can actually measure how learning is changing in this context. So if you know that this is kind of the trick that I'm going to do, and I give you two new images, you actually can probably more quickly find that pop-out effect because you have become familiarized to this. You're more quick to kind of detect where that pop-out is. And so we see this kind of shift over time. So how we do this with EEG is we play about 8 minutes of different sounds. And you can see we have one token that's repeated over and over and over again that we consider our frequent condition. And then we have maybe a tone that deviates slightly. So instead of being a high-pitched tone, it's a little bit lower pitched, which is our kind of deviant model. And then we have these other pieces that are different sounds. So they're not a tone at all. They might be chimes or squelches or chirps. And so this produces a really different effect. And for time, I think I'm going to skip past this video. But you can see our participant enjoying the task right now. So what we look at with EEGs, we say, okay, so at the time that this sound was played, what is the immediate electrophysiological response? And you can see here our grand average time waves. And what I think you can kind of tell from this image alone is how much bigger the P3A as a particular component that reflects attention orienting is in autism. And this is very normal. What we expect to and tend to see in most people is this large deflection, but the fact that we're seeing this as being enhanced in autism is a really important piece. So if we come back to those two rare genetic groups that I mentioned, so we have our GRIN2B group here and our SCN2A group as the second panel with an idiopathic autism group that does not have any known rare genetic conditions and a neurotypical comparison group that also has no known genetic conditions. And what we see is that this lack of typical P3A orienting, and we see this also in a similar speech attention task, is different across these groups. So we see it as being kind of strongest in the idiopathic autism group, and much more reduced in SCN2A, and we see a lack of effects in this GRIN2B group. So what does this mean? Well, it's starting to show us some of the mechanisms of attention that are present in GRIN2B, but we're seeing that attention signal in SEN2A. And this is consistent with other work that I haven't been able to show you where we're linking atypical brain structures in GRIN2B. And it's potentially a really important first target. And the fact that the SEN2A group was differentiating between these sounds, it still might be atypical if we think about the overall kind of process of learning. So we tend to want to understand more about this learning in that context. So what we do is we take this kind of heightened effect that I mentioned before, and what we can do is actually look to see at each time you hear these different sounds, how are we seeing signals change? So these are just two different examples, and you can see these black lines are relating to each trial that the child heard those sounds. So we can take that peak point for each of these different trials and plot them over time. And what we've previously shown is that in neurotypical controls, you see this really, really rapid habituation. So that kind of pop-out effect very quickly like kind of stabilizes to a baseline. Whereas in autism, we call this a stickier attention response, where we're seeing slower habituation to these different novel sounds. And then when we looked back at our GRIN2B and SEN2A patterns, you can see that there's no learning patterns, that the GRIN2B group is very stable across the entirety of the experiment, whereas the SEN2A group is showing this much slower and steady habituation that maps more onto that autism response. So we go back to our little example. Now we can kind of feed these things in. So we're seeing these opposite effects in SEN2A and GRIN2B related to kind of the overall P3A effects, which you can kind of see here as well. But what I think is really cool is then we also have these different atypical habituation effects. So that habituation in SCN2A is actually faster than our idiopathic autism group. And so this is great. So we think about trying to understand and solve one person. Well, the idea would be that we'd have these kind of features that across all of our individuals with GRIN2B, we'd see some similar shared phenomena. And within SCN2A, we're going to see different shared phenomena. And what's exciting for us is then saying, well, if you have an unknown etiology, but they're showing patterns that are more similar to SCN2A, then that creates a shared mechanism or a way in which you might think about proposing different pharmacological targets, for example, or other intervention. And this is great. So I showed you these two biomarkers, but one of the things that we also recognize is that these are just one, and we could be collecting many, and then linking these to maybe other pieces and understanding more about the neural circuitry, maybe using fMRI, and then perhaps using gaze biomarkers, using eye tracking to really kind of flesh all these pieces out, and even to the point of potentially using blood as a biomarker or other kinds of tissue to try to understand the role of these mechanisms. So just kind of as an example, here we're really talking about combining biomarkers across different pieces. So using this P3A response that we found in SCN2A, but there's actually other things that we didn't talk about. So I only talked about that one effect, but there's also low early detection in SCN2A, and we see some ongoing cognitive markers that I haven't described. So now this one biomarker could become three. And then if we link it back into some of the work that we've done with eye tracking, we now are starting to kind of build more understanding. And the big piece that I try to teach all of our trainees and we try to challenge ourselves is then linking it back to that neuronal level. We're never going to measure individual neurons in our work, but we can call back to what our friends in basic neuroscience are doing. And there's some really fantastic work trying to understand how attention signals here as kind of vestibular ocular reflexes, how those are different in SCN2A. And in the animal models, it's a lot more, well, it's easier to understand and to look to see how different treatments are going to change brain plasticity and brain development. So that's an exciting first step. So we also wanted to think more about, well, can the animals also tell us more about our kids? So I've been doing this work with Dr. Annetta Schenk and Laura Block, using fruit flies to try to understand whether or not these human attention habituation phenotypes are mapping back on to the animal as well. And if the flies can actually tell us a little bit more about what we're seeing in our kids. So they're using these Drosophila knockdown models. And this is a video of Laura. What she does is she puts each fly into these different tubes. And to assess habituation, we turn off all the lights, and then we basically strobe this annoying light that the flies don't like. And what happens is at the very beginning, they're startled and respond in a big way. And so they start to jump. But after a set amount of time, and here's like kind of some of the smooth responses, they aren't jumping as frequently because of the fact, that they've learned that this is not really going to harm them. It might be aversive at first, but it's not something that's going to harm them. So what we did is we looked at flies that had different knockdowns in the genes of our kids that we have seen and put them all together and looked to see, well, which flies are performing in similar ways. So we had a set of flies that worked in what we call the severe phenotype, where they actually kind of habituated much earlier than the control flies that did not have any sort of knockdown effect. And you can see some of these genes here. I've mentioned SCN2A before, so this is actually very similar to that SCN2A response. Remember how steep it was? So we're showing that kind of consistency across fly and human here. We had one gene that showed no difference. So both the KISS knockdown is related to CHT8, and we saw similar trials to habituation in both the flies that are control and the mutant flies. And then we had this atypical response, which is more similar to our sticky attention response. And here we're showing the Norexin fly, but this is also where we see our Grin2B kids as well. So the NMDR2 flies are related to the Grin2B gene. So just kind of coming back to think through this, just to kind of showcase, I'm going to do this pretty quickly just to show the fact that if we look then and we take, okay, say the flies sorted our people and then put them into these different groups. And what you can kind of see is that we're showing this effect in each of our different subgroups. So the flies were pretty accurate in saying that the kids who have an atypical habituation effect where it's really sticky are also showcasing that sticky effect. whereas the non-affected had that much more neurotypical pattern of fairly rapid and typical habituation. So we think that that's really exciting. And to kind of further specify this a little bit more, we wanted to I want to show you just at least one example of those genes. So specifically looking at that GRIN2B, and just to tell you ahead of time that we are seeing this kind of group level habituation response. So here's what the GRIN2B group looked like specifically, a little bit more zoomed in, where we're seeing that kind of convergence, where we're seeing a lack of habituation in the GRIN2B group, and the flies were slower to habituate. So it's not necessarily a one-to-one, but we're starting to see this kind of correspondence and conservation across species, which I think is really exciting. So what does this really mean? Like, how would we use this? So there's this idea that if we can map on habituation and more or less a one-to-one experience, we could use the flies to kind of understand what this severe habituation means versus our atypical group and our kind of non-affected genes. And then it's really about using this information to understand what we're trying to treat. So with sticky attention, it might not sound bad, but what we want to do is kind of have a capacity to use other resources. So if we're stuck on information that's not relevant to us, we might need to filter that a little bit better. So we could potentially use habituation to understand how different kinds of interventions are working to improve our brain's capacity. So lastly, and somewhat quickly here, I want to just kind of walk through the fact that we need to be thinking about different maturational and developmental pieces. So a lot of the work that we do is looking specifically within a person as to how they're experiencing information in those 8 minutes, and using that to understand capacity for learning and understanding some of those contextual changes within a person. But we also spend a lot of time bringing kids back. So here's just a kind of a scatter plot. Each row is a person, and to kind of showcase the fact that we're following these kids up over time as ultra rare conditions. It's really important to understand their own maturation. And particularly, we're interested in understanding how those shifts might change during puberty. Then lastly, we in my group take this developmental cascades perspective. So the reason why we study attention is because it's something that's really low level, but we think has really important implications on later acting levels that are necessary for social communication. So if you're not able to attend in a reliable way, then you're not going to be able to learn language as quickly. And so trying to understand some of these low-level systems as a way to understand the mechanisms that are maybe interfering with these higher-level cognitive systems is an approach that we take. So just to kind of walk through, we have some major expectations for what our biomarkers should do. And a lot of this are things that we have direct conversations with our family groups with. So what is it that they're trying to see? And a lot of them are talking about communication changes as being an important goal that they're working with their child around. So to that end, we really need to be developing things that are going to be rigorous and at the individual level be able to kind of reproduce similar responses. So using some test-retest validation of the fact that we're seeing a stable signal, and then understand the natural history, particularly at some of those key developmental pieces. And so having those conversations, I think, not only needs to be had with us as data scientists, but also with these communities to truly understand what are the needs, but then what kinds of categories, remember that list of different biomarkers we could be developing. Understanding when and how these biomarkers should be able to translate, and then thinking about cost and how we're working together. So none of this work can be done with just one lab group. As I mentioned, there's very large scale groups that are doing this across large populations. And to this extent, kind of boiling down to maybe this last piece, we need our biomarkers to be inclusive for everyone. So it's not going to be helpful if our biomarkers only work for boys or only work for girls or only works for those in the South or those in the US. We really want to be thinking about creating inclusive and scalable markers for all. So I wanted to spend the last couple minutes walking through how we've addressed this lack of inclusion in large-scale biomarker development studies, and share just some tidbits of how this is possible, really. So I mentioned these amazing ongoing projects, ABCCT and the EU AIMS. But a lot of, particularly the early parts of those studies, have had really strict IQ cutoffs or maybe adaptive skills. So requiring kids to be able to use a button box to make a response. But when we think about particularly our rare genetic individuals, we tend to see that these kids have co-occurring intellectual disabilities. And so communication difficulties can be a huge barrier. These kids also tend to have very extreme sensory sensitivities. And even thinking about how we work to develop what we're testing and at the paradigm level has been something that we've spent a lot of time thinking about. So, but I want to really focus today on thinking about the context of how we are collecting this data and encourage you all to think about what mobile testing opportunities look like outside of the lab. And I want to do this by just kind of sharing a little bit of this unwieldy at sometimes project that we've created called the BioGene study. So I think we have over 300 or so individuals who have been enrolled in this study to date, with probably over 400 data points, because we've seen many of these individuals multiple time points. So I'm listing some of our funding here. We've seen a lot of different kind of gene groups, and this isn't even exhaustive right now, but we're continuing to meet with each group, and they all have a little bit different needs. So remember, I mentioned kind of opportunities for biomarkers, and we spent a lot of time thinking about these basic attention as well as learning pieces. So for us, what we've developed is a battery that focuses on being inclusive. And that's made us rely on auditory data and tasks, because we've found that our kids just will not visually attend to the screen in a way that some other biomarker projects have been able to. We also have it be as quick as possible, because our kids don't necessarily always last for an hour testing session. So what this ends up being is a standard battery where we at least usually get at least 5 minutes of a resting state, where the goal is to have our kids be still and chill to understand kind of overall brain organization. But we've also been collecting the auditory oddball that I described in the past and a couple minutes ago, and using the kind of these single trial analyses to track habituation markers. We also have, as I alluded to, a speech version of this. And then we've actually started some new adaptations that are really exciting. So we have an emotion regulation task that can be completed in 3 minutes and has some really striking findings. I won't have time to talk about those today, but have me back anytime. And then we also are starting to do some hyperscanning, where we're recording EEG with a proband who has a known genetic condition and their sibling. So stay tuned on some of those pieces. So in this kind of goal towards getting biomarkers, one of the most difficult things that I've maybe kind of alluded to, but didn't really emphasize, is that these are ultra rare conditions. And we're talking about having a couple of hundred of people in the world that have known loss of function variants in this way. So when I set out to develop and start the BioGene study, I was really excited. I had received funding from two small but mighty, let's say, foundations, the Families SCN2A Foundation and Grin TB Foundation, and was super excited. And then COVID hit. So I did what I thought was best, and packed all of my equipment in these 2 suitcases with a paddleboard for scale in this wild little car, and decided let's drive and go all around the country and see our kids. So I started this in fall of 21, and I only had 12 families across both genes that had emailed me and set up a specific date and a time and were committed and had enrolled in the study. So it was really with the community leaders that and the parents themselves of those first early families who posted their experiences to social media that this came into fruition. So I ended up traveling all across the country in my little car in 3 months' time and saw over 55 kids. And you can see some of them in this video here. And it was remarkably successful. Let me turn off the volume on this. So this is just to showcase I did a lot of practicing. So when I could get to a home, I was just me in the car by myself. I could be as quick as possible. This was still fairly new post-COVID. And I wanted you to just kind of get a sense of like how varied these different experiences were. So here we have someone at the kitchen table, someone in their stander. This kiddo at the top here, this was taken at 5 in the morning because that was her happy time. So I woke up and got to the house at 4 o'clock in the morning, which was not my happy time. We had kids that had a lot of mobility assist. Most and many of our kids had limited verbal communication skills. We had some kids fall asleep. We had lots of help and assistance because, again, it was just me. So this is brother. We had pancakes early that morning and really just had a tremendous amount of community support to make this a success. And it didn't always go well. So you saw this kiddo in a video earlier, and then she's done this before, and I showed her a picture of herself, and she was not as happy. But I'd like to point out the fact that she quickly regulated, and after the session, she asked to put the net on and off and on and off and on and off. So while mom did forms, we applied the net, I think, over 15 times. So it was a lot about learning how we have to adapt to what the needs specifically are. We had some kids who were so anxious about this, which is not uncommon for EEG. You can see me in the back running this whole session with an EEG net on my head because that helped this kiddo kind of normalize the process. We've done a lot of this work. Here's a teddy bear wearing the net for the same reason. We had one kiddo who just didn't want to sit. So we stood with them the entire session, which was fine. So all in all, we had a remarkable amount of success. And down here, I just want to highlight the fact that almost every single person completed at least one experimental task. And if we look at the numbers here, the last two columns here from the road trip specifically, of the kids that got the net on, we had over 90% completion for the whole battery. And then when we look at how much data is retained after cleaning and dealing with artifacts, we still have a tremendous amount of data. And what's I think you can kind of see relative to the idiopathic autism group here that was collected in lab, this is a much higher rate of data retained on the road than in the lab. And I think that that's for a lot of reasons, specifically because we are in people's houses. It was a comfortable spot. And it also came down to the task that we decided to use. So we just completed our first European venture where we worked with 15 participants in 2.5 days. And you can kind of see what it looks like when we go to a lot of rare genetic conferences. So we have a whole team. This is showcasing our whole work where we pack our system and hide it as best we can and set it up and go. So kind of coming back to our takeaways and walking through kind of what our expectations are, I think it's really about thinking thoughtfully about what we're doing with our biomarkers and what we think they're going to be able to do for our community groups and making sure that they're mapping the community needs as well. And so to kind of close out, I just want to give you some quick strategies of how we're thinking about this from like a translational perspective. So hopefully one of the things I've emphasized is that we have to be working with people in other disciplines. And this might strike you as being an obvious thing, but it's not always easy to have those conversations. So making sure that you're sharing a language across these different disciplines and using language that makes sense to you. And then also it helps to bring it back to the families and making sure that it makes sense to them as well. So part of this is making sure that our trainees and ourselves have access to these different disciplines. I think that this talk series is a fantastic opportunity. Like you're basically already like selling this point for me, but then thinking about maybe other ways, and that might be writing papers with folks and or going to different conferences, like going to your colleagues' conferences that you don't traditionally attend, and thinking about maybe primers or common resources that can be used more readily across these different groups. And then I didn't have time to emphasize this, but one of the other things, and I think one of the important pieces for us about inclusion is also making sure that we're meeting the needs of all people, regardless of things like their hairstyle. I'm going to leave these tips on here for a second, but just kind of showcase some of the media posts that we shared with our kids here in the South, and you see how thick some of these locks are and how quickly the neck goes on, and our little strategy for pulling locks out between the webbing, and working with things like puffs, to really make sure that we're creating measures and not turning anyone away just because of who they are as a person or what their hair might look like. So I think that's the end for me. I'm happy to answer questions. Just really want to emphasize how grateful we are to our families and our participants. We spend a lot of time traveling with our EEG system to make this work, and are pushing as hard as we can with as many collaborators as will let us, on making this a success. So thank you for your attention and happy to take questions.
Caitlin, I want to applaud you for getting in the miles that you put in to go around and collect all your data. That was impressive amount of literally road work that you went and did to go gather your data. That is huge. Most people, they go between their office and their bedroom every day, and you were probably in countless hotels and diners and truck stops along the way. I can only imagine.
That is absolutely accurate. I should plot the number of oil changes I had along the way. It
That's
was
right. Just how much gas you used on the way. And I have to say that that first little baby you showed, you're putting the little EEG net on, has to be the cutest EEG test subject like ever in the world. Baby is adorable and didn't seem to mind at all
my
about.
very first dissertation baby too. It was the perfect moment.
That's fantastic. And I was really glad to, see that your collaboration with folks at the University of Washington. I was just there last week, and visiting with the folks in the Department of Psychology. And I've known Sarah Webb for years and Ralph Bernier for years. And so it's really nice to see you collaborating with them, as have we, and they're outstanding. I wanted to ask you about the notion of biomarkers is always such kind of a loaded term, and you did a really good job of kind of describing what that kind of looks like. Everybody sort of uses that term. It's like one of those things where, like in The Princess Bride, where the actor says, "You keep saying that word, but I don't think you know what it means." And I'm just kind of curious about when people say that term, what do we, especially in autism, what do we want them to understand about autism where a biomarker is really meaningful, and is something which has reliability, reproducibility, and has specificity for what we're looking for, so that people don't just start, oh, I found some statistical result, it's a biomarker. Well, is it, or what else are we looking for? And do you have any advice?
I think that's a great question and point. So, and literally, I just came from my lab meeting where we had this exact conversation about like, what do we really mean? Why are we doing neuroscience was the prompt that we were addressing in lab meeting, which is kind of an existential crisis sometimes. But I think for us, one of the things that we've been pushing back on is that it's on the onus is on the researchers to define that well. And I think you're absolutely right. It's got to be more than just a statistical correlation. It has to be something that we expect is stable at baseline, but is likely something that could be potentially changed as we see intervention success, because a biomarker that doesn't move to our clinical interventions is not necessarily going to be particularly helpful. So having it be kind of something that captures a wide amount of variance, but like maps that back on to the behaviors. And I think one of the things, and this is why I love I use my little pyramid all the time, is that sometimes the biomarker, it should hopefully connect to the human phenotype, but it could also be the case that it connects really closely with what we're seeing at lower levels that may be more specific to kind of molecular or cellular levels too, and understanding how that could be important. It can also be leveraged as a way to kind of think through why certain therapies work and why others don't. I think just creating a biomarker to say this is autism is not something that we're at least trying to do. We really want to be developing biomarkers that have this clinical utility. But we're not naive to the fact that we're pretty early in some of this work. And I think when I was thinking about what to share here, I didn't go the other direction, was to talk about the fact that because we work with all of these different genetic etiologies of autism, we're using some really important data-driven computational tools to try to understand where is there shared mechanisms in a wider set of kind of the populace. Some of that work will be probably be coming to fruition in the next like 6 months or so for us. We haven't finalized the data push or the analytic push rather. So I think that there's these like multiple levels of being mindful and thoughtful. And if our biomarker, like our P3 biomarker seems pretty reliable, we're really excited about it, but it probably is never going to work in isolation. I think it needs to come together and be tethered to a behavioral or clinical phenotype specifically, and then also potentially be linked to other, these kind of idea of combinatorial biomarkers is going to be incredibly important.
Oh, absolutely. And I'm curious about when what you just described, it seems very similar in spirit to the RDoc effort from a few years ago, which I'm still a big fan of, but I'm not quite sure that people may understand what it means and what its current status happens to be.
Y- Yes. I think RDoC is tricky because um, every level seemed to kind of exist in isolation. And I think that it's tricky to understand how to link across them, right? Because it requires you to measure multiple things, or as what we've been trying to do, to reinterpret how you get multiple levels of information in one data set that can answer potentially different questions. So this is another reason that I tend to really like ERPs, and I kind of mentioned it but didn't super get into it, that this idea that ERPs tell us the whole cognitive story,
Mhm.
or as much of it as we can, because we have such a fast acting, we can measure at the millisecond level. So when your brain is integrating and detecting and coding and reasoning about information, those are each discrete ERP components that we can derive and understand and really look to see across the whole head how those are telling us the story about how information is received and then like represented, and how and why we act on information. And so to me, that might be a way of better kind of linking across those classic domains from this RDoC perspective of maybe using what we have, but thinking about it in that integrative sense.
I liked the connection that you made between the work you're doing in Drosophila and the genetics of the two different kind of groups you had. I think one was the GRIN2B and the other one was the SCN2A. And I was curious about those, is that one in the experimental paradigm that you have with the strobe light, and the startle reflex and the jumping around, and I mean that's pretty clever. I'm wondering if how did you choose one? Did you choose a particular frequency of the strobing that you knew would be maximally startling for them? And or can you like sweep across different frequencies, or can you inject another frequency of strobe in there to kind of restartle them even after they've habituated to an initial startle? Does that make sense? Because maybe some autistic fruit flies, if there really are such things, might startle differently or have a different habituated response. I'm curious about that.
Yeah, that's usually I didn't talk about any of my limitations, but to me that's one of our huge limitations, which is that we're talking about habituation in two different modalities. So we're looking at auditory habituation in our kids, and the flies is a visual piece. And there's other elements of their fly battery. So it's a steady state. So it's cycled at a every four uh maybe it's every second. I'd have to double check the battery, but they have a steady kind of response. And because we're cognitive neuroscientists, we have a jittered intertrial interval so that you don't build those predictions. So empirically and from like an experimental design perspective, we actually have very different tasks. And so in some ways I'm like, well, okay, that's cool though, that despite those potential confounds, we're seeing some shared pieces. But what we really want, and we're looking for some funding to kind of have be able to support a student who can go in and do our auditory task with the flies, right? And then have on our side be able to do the light habituation and to start to see that alignment more closely and more specifically, even in control flies and control participants. The other piece is that as a translation, I consider myself a translational cognitive neuroscientist, and um, I spend a lot of time sitting and talking with my rat researchers and mice researchers about what they do and developing those tasks from the get-go to map more on. So there's a lot of like aversive learning that we do in animals that's fantastic. Like you can feed them pellets and you can shock their little feet. I'm not going to shock our kids, and
I hope
um,
not.
yeah, and we do sometimes feed them M&Ms, but it's not a salient motivator. And there's these like tricky pieces where really having conversations about how to align at the design level, I think is going to be increasingly important, particularly because I think we're going to make the fastest progress towards pharmacological interventions using these animal systems, but knowing what that means in the for the humans has to come from our cogneuro perspective and our behavioral perspective. So we need to work together, but they can run, a whole set of hundreds of flies in a matter of 30 days, where it takes me 2 years maybe to see 100 kids, right? So there's interesting limitations. And so trying to identify those, but then maybe make use of them and in smaller like sub studies, doing some of that validation work, I think is going to be absolutely necessary and exciting to kind of inform what we know.
In our last few minutes here, I wanted to ask you, what role can people who are the data scientists, the deep learning specialists, those people delving into AI and other kind of statistical methodologies that are kind of like really advancing, like every day we're seeing new stuff come out. How can they play a role here, and at what level are they the most helpful for your work? And how could you be helpful for them to understand how best to analyze these types of data?
So I work with a lot of biomedical engineers in different ways. So one is like Dr. Nicola Pini, who's a biomedical engineer, comes with me on these research trips and is doing some of the data collection. But what I think that your question is getting down to is this idea of using existing and emerging data sets in smart ways. So I sit on a lot of students' dissertation committees who are using like machine learning approaches and trying to understand what the best machine learning approach is. And it's not that we need to have some sort of consensus. I think that that's the thing we need to remember, that like what is common across all, and how can we use that to kind of accelerate what we know and how we're going to treat our kids. And then I also think that using, and that's what we're trying to do, is putting in all of our biomarkers into models and saying, here's what we're seeing. And even like to the level of resolving the spatiotemporal opportunity, to kind of understand across the scalp, which refers to all the sum of all of the brain activities at any given millisecond, and using that to really kind of drive what we are seeing. We are going to need a computational approach. So that might be kind of collapsing all of these existing and emerging data sets, like I said, and trying to see what are we seeing as data-driven clusters, and what are we seeing as kind of like confirmatory clusters, right? Like, does it make sense to do this genetics first approach? And I can tell you some of these kids look really different, even though they might have very similar variants. So
Mhm.
how do you kind of assess those pieces? I think we're going to need that computational effort to be able to mine this really beautiful existing data and know what we need to be doing and how we should be focusing efforts on collecting new data.
Well, Caitlin Hudak from University of South Carolina, excuse me. Thank you so much. This has really been a fascinating and fun tour through your work that you toured the U.S. to give. And on your next road trip, we invite you to come by Charlottesville and visit us. And I hope you will. Thank you so much. Thank you, everybody who's joined us and is listening wherever you are on the planet. And we will see you next time. Thanks again, Caitlin.